Dr. Krueger is currently the Associate Vice President and Technical Director of R&D at LabCorp, one of the largest reference laboratories in the United States. He has a tremendous experience in molecular technologies, high throughput sequencing, and lab automation, and has worked extensively in the area of infectious diseases. He has spearheaded the effort that gained LabCorp the first commercial laboratory FDA EUA clearance for COVID-19 testing here in the United States. And the title of Dr. Brian Krueger's talk will be Development and Scaling of the LabCorp COVID-19 RT-PCR test. Dr. Krueger, welcome, and I'm going to turn the podium over to you.
Great, thank you, Carl. I appreciate that fantastic introduction. It's very nice of you. And also want to start off by thanking BioInsider for inviting me to speak about this important work that we've been doing here at LabCorp to diagnose patients with active COVID-19 infections. I hope that by the end of this presentation, you really get a feel for how much effort went into developing and scaling this assay to meet the national demand for a diagnostic assay to detect COVID-19. This involved many, many weeks of development work, working directly with our vendors, the FDA, both to validate the assay and also assure the supply chain of the reagents for backup reagents, making sure that all of these things were available so that we could continue testing during this very challenging time. So just a quick development timeline. I'll come back to this slide a couple of times throughout the talk. First cluster of cases was initially reported at the end of last December, and by the beginning of January, it was confirmed to be a novel coronavirus, with the genome of the virus being released by the Chinese government on January 12th. Approximately 2 weeks later, the CDC published its original primer and probe sequences, along with a protocol for running the assay at state health labs. So when the CDC protocol came out on the 29th, we at LabCorp, we immediately decided that if COVID-19 was truly going to become a pandemic, that we needed to provide a much, much higher throughput solution than what the CDC had proposed. And in doing so, that meant that we would have to pursue our own emergency use authorization from the FDA and all of the additional testing that would entail. So on the 30th, we ordered all the CDC probes and primers, along with beginning the process of tracking down the libraries of interfering respiratory organisms. When the CDC was finally approved for their test on February 4th, we notified the FDA of our intention to submit our own EUA for an internally developed assay, and continued the process of tracking down all of the additional interfering organisms that the FDA was then requiring. The longest lead time organism, of course, was COVID-19 itself, and it wasn't made available until February 28th. And we had intended originally on validating on a synthetic target that we had designed at IDT so that we would be able to provide testing much sooner, but we were told that that wasn't acceptable, so we had to wait for the virus to finally be made available. We completed the validation of our assay on March 4th and launched a manual version of that assay into production on March 5th. This was following the new guidance from the FDA at the time that high complexity labs could launch their assays prior to approval, as long as they made it known that they intended on submitting the validation data. So on March 16th, we finally got the authorization for our internally developed assay. It was the first, and we were the first commercial lab to receive that authorization from the FDA. So the LabCorp assay, along with many of the other nucleic acid-based assays that are authorized by the FDA, is based on PCR or reverse transcription PCR. The assay uses a one-step RT-PCR, where the reverse transcription of viral RNA and then the subsequent PCR amplification occur in the same reaction mix. This simplifies the detection process, and it also streamlines the assay for high throughput implementations. So just a brief overview of how this all works. I'm sure a number of you understand this already, but I'll go over it very quickly. In the first cycle of an RT-PCR reaction, the RNA template is used to generate cDNA. This is followed by normal cycling of PCR out to about 45 cycles, of which only 40 cycles is analyzed. Target detection is determined by the binding of a fluorescently labeled probe to its target, and then if it's bound to its target, the fluorescent tag is released from its quencher by the 5' to 3' exonuclease activity of the DNA polymerase. This process continues cycle after cycle, of course, and allows for the quantitative detection of DNA in the PCR reaction. As I said, the LabCorp assay is modeled after the CDC assay, and it targets the nucleocapsid sequences of COVID-19. N1 and N2 are COVID-19 specific. N3 is pan-SARS, and will detect SARS-CoV-1. Human RNase-P is used as the internal control to ensure that all samples have extracted nucleic acid in them. And each probe is labeled with FAM. So in this original implementation that the CDC proposed and that we started and launched on, required 4 separate PCR reactions. So one for each target, since all of the probes had the same fluorescent label on them. As I said, the CDC assay was granted its EUA on February 4th. They launched on the QIAGEN biorobot for extraction and the Thermo Fisher 7500DX qPCR system for detection. These are both very, very low throughput instruments, and they're not capable of the testing volumes that were going to be required for adequate detection of infection during a pandemic. We recognized this really early on, began development of a very high throughput implementation of the CDC test. For this, we used the Thermo Fisher QuantStudio 7 Flex, which is capable of performing qPCR as either a 96 well or a 384 well instrument. So we really like the flexibility of that instrument because it provided us that flexibility to launch very quickly originally as a manual 96 well assay, and then rapidly transitioning to much higher throughput 384 well based versions, both of which were in 384 well plates. One of them we refer to as single plex, and the other a multiplex, which I'll go through in greater detail in a minute. Extraction was developed on a number of different systems, a number of different high throughput systems from Roche, Thermo Fisher, and Hamilton Robotics. And each of these can output approximately 96 samples per hour. Because we were not using the exact same instruments and protocols as the CDC for our implementation, LabCorp had to pursue an independent emergency use authorization from the FDA. The EUA process is a rapid emergency process that's used by the FDA in times of crisis to allow tests to be approved without going through the entire 510K de novo process. This is a little bit like 510K light, and not all that different from what labs generally do when they develop lab-developed tests. But this process differs from a traditional 510K submission, mainly in the scope of the validation that is required. Labs still have to do limit of detection, inclusivity, exclusivity, and perform a clinical evaluation, but the scope and the scale of those validations is significantly truncated, which allows for much more rapid approval of the test through the FDA. The results of the LabCorp EUA submission are summarized on this slide. Limit of detection was determined by running 20 samples at each concentration, and the LOD of the assay is basically where 19 out of 20 samples returned positive. Exclusivity testing was performed against 26 required respiratory organisms. There was no unexpected cross-reactivity. For the final clinical evaluation, we tested 177 negative samples and 96 positives. all the negatives were negative. 16 out of 16 of the clinical positives were all positive. 79 out of 80 of the contrived positives were positive. We had one single contrived sample at the LOD fail, which is that 19 out of 20. And the final LOD of the assay is 30 copies per reaction, or 6.25 copies per microliter. And I really think that Dr. Hilliard's talk earlier this morning is instrumental in explaining how limits of detection actually apply in a testing environment. It's a little bit disingenuous to extract and extrapolate copies per reaction and copies per microliter LODs from individual EUAs as a method of doing a comparison of the sensitivity of assays in the field. We're LabCorp. We run a number of different SARS-CoV-2 assays here. So we've also done these comparisons internally about the sensitivity of these high throughput assays that are available on the market. Here I'll just show a little bit of data comparing an assay with an advertised LOD of 100 copies per mil, and then our EUA. The CT crossing points between these assays are almost identical, with our EUA having about a 1 log edge when the sample is run through both systems. So I think because of this, it really is wholly inappropriate for competitors to extrapolate sensitivity of the LabCorp assay out to 6,000 copies per mil to make arguments that their assays are more sensitive, because that's just not the way that this is done. I mean, everybody's running qPCR and the sensitivities are all going to be quite close to one another. So as you can imagine, when we launched this assay on March 6th, we had been planning for many weeks in advance to scale the assay to support tens of thousands of reactions per day worth of capacity. Our high throughput automation implementation of the assay launched approximately 2 weeks after the initial launch on March 21st. The first high throughput implementation we refer to internally as Singleplex, is that Multiplex is the 4 reaction assay into a 384 well plate, which allows us to run 96 samples at a time instead of the original launch volume of 24 samples per plate. The high throughput versions of the assay are automated on Hamilton Star liquid handlers, and all of these scripts were developed internally by our automation engineer, Kimberly Wagner. The Hamilton team, of course, was really instrumental in rapidly processing our orders for these systems and installing them across our testing network. But again, it quickly became clear that even that Singleplex version of the assay wasn't going to be able to keep up with the volume demand. So we spent a few weeks finalizing a Multiplex version, which launched into production on April 3rd and received an FDA EUA amendment on the 14th. We had always intended on launching a high throughput multiplex version of the assay, but because of limitations in the FDA process, we launched on this lower throughput manual and singleplex version to begin with. Traditionally, the requirements for launching a multiplex assay through FDA are much more stringent, requiring significantly more validation effort. However, due to the urgency of the crisis, some of those requirements were eventually relaxed. Ultimately, we multiplexed the assay by changing the dyes on N2 and RP, dropping that N3 target at the recommendation of the CDC and FDA because it misbehaved in the CDC assays. We never had any problems with it, but it freed up a little bit of space for the multiplex. This allowed us to ultimately quadruple the throughput of our liquid handlers and quant studios, though. And it also allowed us to clear a backlog of about 120,000 samples over a single weekend. So it was really, really important that we were able to get this assay into production. You'll notice that on the previous slide, there was about a 2-week gap between the launch of the singleplex assay and the multiplex assay. And this delay was really caused primarily because of supply issues from reagent vendors who had very long queues for oligosynthesis. The assay itself was really ready to go when the singleplex automation launched. We just didn't have the the reagents in-house to operationalize the assay. So finally, the latest high-profile approval you've heard from the FDA about regarding us is the approval of our at-home collection kit on April 20th. And as the outbreak progressed, it really became clear to us and others that conventional methods for respiratory virus testing were really inadequate in dealing with the volume of affected patients. By doing this kind of testing in the traditional physician environment, asking a patient to present in person poses risks to them, to the healthcare practitioners and other patients. So one potential method for alleviating these risks while still providing safe and effective testing is for patients to collect their own samples at home and then send them back in. And the Pixel collection kit that we developed and got approved does exactly this. Before a kit is shipped to a consumer, they have to answer a survey online about their symptoms, which is reviewed by a physician before shipment of the kit to the consumer. The kit includes swabs for collection, a saline tube, a gel pack, a return shipping box, and a label. After the consumer collects their sample, they place the saline tube and swab into the return kit, and then they schedule FedEx to come and pick it up, or they can drop it off at a FedEx drop-off location for shipment back to us. This product was first made available to healthcare workers and first responders, but we are now offering it to anybody who wants to visit pixel.labcorp.com. It is available nationally now, except for in the states that do not allow direct-to-consumer testing. The validation of this kit also included an extreme temperature excursion study that was required by the FDA to verify that samples near the limit of detection of the assay returned back positive, even after they sat at 104 degrees Fahrenheit for 30 hours. As many of you have probably seen and heard, many supplies are limiting for this testing in the testing environment. So in addition to the Pixel validation, we've spent the majority of our time internally validating alternate collection devices, transport medias, extraction kits, amplification enzymes. Basically, if you can think about something that is used in the lab for performing RT-PCR, we have validated 3 or 4 alternatives for that exact thing, down to tips, just to try to head off any of these future supply chain issues. And I think this really is probably one of those most underappreciated facts of the pandemic. Testing has not been limited so much by test capacity, but really by logistics and supply chain. Performing this accurate, high quality testing at scale really requires an army of people and robotic systems. And these are things that people just don't think about. How do you kit tens of thousands of swabs a day? How do you get those kits to the consumers? How do you then receive those kits back in the lab, open them up without spreading the virus all over your facility, and then get those samples onto plates for testing? The amount of work that we do is really, really staggering before the sample even makes it to a plate in the testing environment. And I think these are key points for consideration when we're talking about achieving massive single site test volumes on any sort of a relevant timescale. At LabCorp, we're really exceptionally good at logistics and supply chain. We process millions of clinical samples a week for diagnostic testing. And it's one of those key reasons why we were not only able to launch the first commercial assay, but also be the first commercial lab to clear the enormous testing backlog that was created by our really national inability to test patients for most of February and March. At present, the enterprise testing capacity exceeds 100,000 samples per day. We are currently testing at 5 sites across the continental United States, and we'll be going live at others in preparation for really what we and everyone else is anticipating to be another challenging surge in cases here in the fall. However, we're in a good place now. Our capacity currently exceeds demand, which is quite good. It means that we can continue to turn these samples around very, very rapidly. And really, if the map here to the right, tracking the positive samples that we've received over the last few months is any indication, this virus isn't going away anytime soon. And it's going to require the continued national effort of commercial diagnostics providers to track infections and aid in the decisions both at the individual and the national policy level. So finally, I'd like to end by acknowledging a handful of people that were involved with this. The LabCorp team here is just the R&D team. It doesn't include the hundreds of people within the organization that have helped to roll this out to the 5+ different sites throughout the country. And it also doesn't include all of our thousands of technicians that are running this assay every day, 24/7. Ayla Burns did all of the EUA studies for the submission. Kim Wagner did all of the robotics programming. And that's pretty impressive that, you know, these two people are really the workforce behind the 4 million samples that we have tested to date. Also, huge thanks goes out to the team at Hamilton Robotics and also our partners at IDT who have been instrumental in really being able to provide the reagents for us to continue this testing for the last couple of months. Here's a quick look at some references. If you want to take a look at our EUA or our IFU, you can take a look at them here, or you can look them up on the FDA website. And I'm happy to take any questions that you might have.
Dr. Krueger, thank you for that excellent presentation and view into the scaling up and operation and implementation at LabCorp. From the audience, we have a couple of questions that sort of have a sort of interrelated nature. They're both pertaining to the at-home collection test option with the Pixel testing system. One is, what kind of percentage of the testing or testing volumes are you seeing in terms of the in-home testing? That is kind of what's the uptake of individuals performing home collection and sending. And also, during the validation of that testing approach, were you required to demonstrate that the shipping package was potentially contaminated with virus through, you know, the individual collecting the sample, handling the package? So sort of overlaid, what's the kind of testing volume you're seeing or percentage of testing? What's the uptake? And a little bit more about the validation regarding the sample packaging.
Yeah, so I can't comment on specific numbers through specific channels. I'd say that testing for at-home collections, you know, through Pixel or through employers or through our on-site testing initiatives is quite high. It's been really important for us to be able to get out to these sites, hand these kits out to people, and allow them to do this testing either themselves there on site or at home. So I think we're all really proud internally that we were able to make this channel available to people. As far as doing a test to determine if people contaminated the tubes and the reagents, we did do an employee self-collect study where, you know, the employees were observed, but they weren't instructed on how to do the collections. We did not receive any externally contaminated packages back, but, you know, I also would say that we didn't do an extensive clinical study to determine if the packages were contaminated. I mean, I think it's certainly possible that's true, but I mean, it's not untrue that MP swabs or other swabs collected in the physician environment aren't also contaminaminated. So that's why you really have to be very careful when you receive these samples back into a high complexity clinical lab to make sure that you're not spreading these things. And I think there it is good data externally that, so say the patient contaminated the outside of the package, and maybe that's what this question is getting at. Are we putting postal workers or FedEx or UPS workers at risk of contracting the virus? And we have good data that the virus doesn't live very long on these paper surfaces. And generally these packages are sitting around for quite a while before they're handled by anybody other than the person who collected the sample. So I think the risk to package handlers is quite low.
Great, thank you. Perhaps one more question, which kind of has a bit of a relatedness, which is your laboratory personnel that are directly involved in testing for SARS-CoV-2. And this is a question that many laboratories are struggling with, is that what approaches have you taken at LabCorp to sort of assure laboratory personnel safety in this kind of environment? And also, have you implemented any type of sort of monitoring, even testing, if you will, of the actual technologists at the laboratory bench working and doing the SARS-CoV-2 testing?
Yep. Yeah, no, so those are really great questions. And, I think I would say that LabCorp is a healthcare company first. So we put our employees first. And, essentially from day one, we have been doing health screenings before anybody walks through the door. So when you come in to the laboratory, you come up to a table, you must be wearing a mask. You talk to an attendant who takes your temperature, asks you a number of questions about exposure or symptoms. If you answer yes to any of those questions, you're told to go home and take the day and notify your supervisor that you're not coming into work. So that's our first line of defense. If somebody does come back positive, or somebody comes back with symptoms, we offer them free testing internally so that they can determine if they do actually have the virus. When we do have employees that come back as being positive, we do contact tracing internally to be sure that everybody in the lab who was exposed has the opportunity to take a test or be observed or see a physician to make sure that they are not a risk to themselves, their family, or anybody else in the lab. So I think from the beginning, we have taken this incredibly seriously because, not only is an issue for our employees themselves, but if we have an outbreak internally, that puts the entire nation at risk. So we have to be really, really careful about the things that we do internally, because it's not just about us, it's about everybody.
Great. Well, thank you so much
Yep.
for your presentation again. At this juncture in our meeting, I'm going to turn the podium back over to Dr. Emily Lee, who's going to talk about the next speed networking session break, and when the next talk will begin.